Thank you. So, June 2026 was an extraordinary turbulent month for Abivax. First, let me recap what happened during this roller coaster month. On June 1st, the company released Obefazimod Part 1 maintenance data. If we look at efficacy alone, Obefazimod can be described as the best performing once daily oral drug for ulcerative colitis to date. Before the data release, the management guidance was about 20 to 30% placebo-adjusted remission rates. My own prediction was around 40%, and then the results came in at 39% for the 25 milligram group and 40% for the 50 milligram group. From an efficacy standpoint, these results were unquestionably outstanding. Compared with other ulcerative therapies, this study enrolled one of the heaviest baseline patient populations, including a subset of patients who had previously been treated with 4 lines advanced therapies and even JAK inhibitors. Even so, the drug still delivered results that were better than those seen with JAK inhibitors themselves. Because the data were released aftermarket, the stock initially surged from around 130 to roughly 160 following the press release. However, the market's focus shifted swiftly. A handful of isolated cancer cases emerged in the highest dose 50 milligram cohort, and that immediately triggered a sharp aftermarket sell-off. When those few cancer cases appeared, the market reacted like a startled frog. That anxiety quickly turned into aggressive selling across the street, sending the stock all the way down to a low of 77, a drawdown of nearly 40% from the peak. Yet the greatest controversy surrounding the dataset was ultimately not efficacy, but safety. On June 4th, at a conference hosted by Guggenheim, the company provided additional context and explanation around these cancer cases. Some patients already had a prior history of cancer, with others had previously been exposed to the drug that may have been associated with higher cancer risk. In addition, the chronic dysplasia case turned out to be something of a false alarm. It was not actually even cancer, but merely a precancerous lesion. It also appeared that the company may have been overly confident in the data and released a table pulled directly from the system without sufficient processing, clarification, and cleanup. These explanations helped restore some investor confidence, and the stock rebounded modestly. Obefazimod's mechanism of action has never been fully understood from a translational standpoint, and that lack of clarity has long been a focal point of criticism. There has been almost no precedent of biotech company independently running a phase 3 trial of IBD in this scale with over 1,000 patients. As a result, many public market investors were simply not familiar with how to interpret cancer cases arising in an IBD phase 3 trial. Then the management team recognized that the market was reacting less to the data itself than to the fear of uncertainty. As a result, the company made a highly decisive move. Instead of waiting until the second half of the year to release the full Part 2 maintenance dataset, it accelerated the timeline and prepared the data for earlier disclosure. On June 29th, Abivax released the full Part 2 maintenance data. The dataset once again confirmed Obefazimod's historically leading efficacy profile, while also clearing up the market's earlier misunderstanding around cancer risk, addressing investors' core concern and helping stabilize the stock to recover. Abivax then launched a $600 million financing, received $4 billion of demand, represent roughly 7 times oversubscription. In the end, the financing was upsized and closed at $900 million, making it the largest PIPE financing in the biotech public market this year. Next slide, please. So, to understand the cancer signal in Abivax maintenance dataset, we first need to establish 2 conceptual pillars. background cancer incidence in autoimmune disease, and why non-melanoma skin cancer, aka NMSC, is routinely analyzed separately from all malignancy in clinical trial safety reporting. So first, let's talk about background malignancy risk in immune-mediated inflammatory disease. Many immune-mediated inflammatory disease carry an elevated baseline cancer risk compared to general population. This elevation is driven by multiple factors. Chronic inflammation, which promotes mutagenesis and impair immune surveillance. Immune dysregulation inherent to disease itself, older patient age at enrollment, cumulative prior immunosuppressive therapies such as therapy, methotrexate, and other biologics also already showed higher cancer risk, and the higher surveillance that apply to this patient, which tends to increase the detection rates of cancer. In other words, patients who enrolled in a modest to severe active UC trial are not starting from a zero cancer risk baseline. Then how should we evaluate those? There is a major unit, patient years. In clinical trial safety reporting, malignancy incidence is expressed as number of events per 100 patient years of drug exposure, which means either one patient in 1 year observed for 1 year, 100 patients observed for 1 year, or 50 patients observed for 2 years, both produce 100 patient years. Expressing events per 100 patient year, adjusted for differences in follow-up duration and trial size, making incidence rates comparable across studies with different observer periods. So why is this matters? For ABVX maintenance trial ran for 44 weeks of blinded follow-up after an 8-week induction in a patient population with high background burden of prior immunosuppressive therapy. Cancer events observed over a finite exposure window cannot be meaningfully interpreted without this exposure-adjusted denominator. And compared to other diseases and other drugs, for example, psoriasis and other drugs like JAK inhibitors, Obefazimod definitely didn't show higher cancer risk versus others. And another thing is we need to understand why NMSC is analyzed separately. Non-melanoma skin cancer, which composed of basal cell carcinoma and squamous cell carcinoma, is a true malignancy but is routinely excluded from the primary malignancy analysis in trials of immunosuppressive therapies for several reasons. First, for the prognostic profile, NMSC is the most common malignancy in immune-suppressed patients and in general population. It is rarely life-threatening when treated appropriately and has fundamental different clinical course from visceral or hematologic malignancies. NMSC risk is strongly driven by cumulative ultraviolet radiation exposure and total lifetime immunosuppressive burden, not necessarily by drug under investigation at the trial moment. Many Abivax enrollees had years of prior biologic and JAK inhibitor exposure. Another thing to consider is regulatory convention. The FDA and EMA both accept separate reporting of NMSC as a standard practice. Approved drugs across multiple classes, including TNF inhibitors and JAK inhibitors, have NMSC listed distinctly in their labels. So clinical decisions separate NMSC allows clinician and regulator to assess the risk of potential more serious systematic malignancies independently. And the signals most concern that's the real signal most concerns prescribers and payers. Accordingly, when evaluating Abivax safety profile, the immediate question is not the raw number of cancer cases, but exposure-adjusted incidence of serious visceral malignancy of NMSC compared against appropriate disease-adjusted background rates. And so what would make a cancer signal drug related? From a clinical pharmacological and regulatory science standpoint, a drug attribute malignancy signal would typically require multiple coverage line of evidence. First, you need to be access over background adjusted comparator. Second, you need to have a temporal coherent. The cancer should generally emerge after sufficient drug exposure. Very early onset cases in short trial often reflected pre-existing conditions, accelerated detection rather than drug itself. Third, whether the drug is dose or exposure respond. Higher doses or longer exposure should correlate with higher incidence if the drug is causally driven the malignancies. So from some PI interviews, I understand the obefazimod have already been used for Europe for 5 to 7 years for a lot of patients, and there are still some patients on remission and on the drug. And third, the biological plausibility of the drug. If the drug's mechanism of action provide a credible pathway to observe the cancer type, for example, the OX40/40L and the Kaposi sarcoma in atopic dermatitis, then the correlation should be more clear. None of these criteria was rigorously met in initial Abivax maintenance dataset discussion, which is a key reason why the market reaction was considered an overreaction. The next slide, please. Then, with the solid understanding of the background, let's see how obefazimod real safe data looks like. Abivax announced a highly anticipated part 2 of 44 weeks maintenance data. They evaluated obefazimod, and there are two non-MNSCs. Two are reported in part 2 and both drug unrelated and attributed to pre-existing risk factors, resulting in 0.48 event rate per 100 patient years, which falling within mid-low range guidance of 0.3 to 0.7. On NMSCs, total for NMSC, 2 on 55 milligram and 2 on 25 milligram and 2 on 50 milligram, 2 basal cell carcinoma and 2 squamous cell carcinoma were reported in part 2, resulting in 0.95 event rate per 100 patient years, which also fall in mid-lower range of the guidance of 0.7 to 1.4. Importantly, the balanced NMSC cases across both doses suggests there is no dose-dependent impact of obefazimod on NMSCs. The two non-NMSCs, one is MPN, which is myeloproliferative neoplasm, had a history of thrombocytopenia since 2023, elevated platelet at screening, and the 3 prior UC treatment, which were Humira, Remicade, and mesalamine. And the prostate cancer patient has history of limited adenocarcinoma of the prostate, multiple prior UC treatments, which are Remicade, Entyvio, and the history of azathioprine usage. So those study investigators conclude that both cases were unrelated to obefazimod treatment. 2 cases of basal cell carcinoma and 2 cases of squamous cell carcinoma spread evenly through different doses and were largely influenced by therapy usage, medical history of skin cancer, and 2 prior advanced therapies. The 2 integrated dataset across phase 2 and phase 3 induction and maintenance data showed 0.59 event rate per 100 patient years across 1,704 total patient years, which is well below the guidance of 0.7 to 1.4. Next slide, please. Obefazimod achieved a higher endoscopy remission in induction non-responders than Rinvoq achieved in induction responders, which had previously held a record for the highest endoscopy remission rates. The trial has the worst patient baseline ever and the best efficacy ever. This is arguably the greatest non-responder crossover efficacy, everything, and this is hugely important for commercial uptake. In other words, the non-responders ultimately ended up with higher remission rates than the responder of what was previously considered the most efficacy drug on the market. This showed the induction only treated patient for 6 weeks, but now Abivax has demonstrated is that if patients are simply treated for longer, a substantial portion of those initial non-responders ultimately do respond. This is massive for long-term revenue generation, which the result is also aligned for phase 2 and OLE data. This is not only the first in class or best in class, but the best ever in disease drug for ulcerative colitis. In summary, I think I'm highly encouraged. With the safety overhang now largely behind us and the broad consensus supporting the pursuit of both maintenance doses, I think Obefazimod will become one of the most valuable assets in IBD space ever, with the potential to become a cornerstone oral treatment for UC. That's pretty much. That's